All right, welcome to the Explainer. Today we're jumping into a source that's asking some pretty tough questions about a scientific tool most of us have heard a lot about, the PCR test. Okay, let's just dive right in with the claim at the very heart of this. It's a quote our source attributes to Dr. Kary Mullis, and yeah, that's the guy who actually won the Nobel Prize for inventing the PCR test. He apparently insisted his own creation was never meant for and should not be used for diagnosing disease. I mean, that's a pretty explosive place to start. So that quote right there, that's the whole setup for what we're talking about today. We're going to break down the argument from this source that this brilliant Nobel-winning tool is being used in a way that its own inventor might have warned us against. So here's the core idea, the real heart of the argument. It's this contrast between what the test was designed for and how it's being used now. The source says it was made for the clean environment of a pure lab, but now it's being used on dirty samples from the real world. Think about it. One is analyzing pure, isolated genetic material. The other is trying to find a tiny signal in all the noise of a clinical swab. Okay, so to really get what they're arguing, we've got to understand how the source explains the PCR process itself. What is this thing actually doing?
At its heart, the source basically calls PCR a kind of genetic photocopier. I love that analogy. It takes this tiny piece of DNA, or RNA, a sequence so small you'd never see it on its own, and just copies it over and over and over again until bam, you have billions of copies and it's easy to spot.
So it works in these cycles, right? First, you get the genetic stuff out of the sample. Then you add these things called primers. You can kind of think of them like a search command in a document, looking for one very specific phrase. Once it finds it, an enzyme comes along and builds a perfect copy. And then you just repeat that whole process, doubling it every single time. 2 becomes 4, 4 becomes 8, 8 becomes 16. You get the idea. It grows exponentially.
This brings us to what might be the most critical piece of the puzzle here, the cycle threshold, or CT value. That's just the number of times you have to run that copy machine before you get a hit. And here's the kicker. The source points to research that says for a virus like SARS-CoV-2, if you have to go over 24 cycles, the original sample probably wasn't even infectious. But, and this is a big but, it claims many labs were running tests at 40 cycles or sometimes even higher.
And that whole issue with running too many cycles? Well, it feeds directly into the source's biggest worry, the risk of what it calls false positives. Now, these aren't false in the sense that the genetic code wasn't found. They're false in the sense that they might not mean anything clinically. So from this viewpoint, that CT number, that context, is absolutely everything.
To really hammer this home, the source uses this brilliant crime scene analogy. It says, to get a solid DNA match for a court case, you need something like 35 to 50 unique genetic markers, right? You need to be sure. But the source argues using one of these PCR tests for a diagnosis is like trying to convict someone based on just one common marker, one that tons of people might share. You can see the problem, right? And why does that matter so much? Because a real-world sample from a swab, it's not clean. The source has this great term for it. It calls it a microbial soup. I mean, think of what's in there. You've got whole viruses, sure, but also bits and pieces of dead viruses, other bacteria, and maybe even contamination from the air. It's a complicated mix. So, that leads to this little thought experiment the source poses to make its point crystal clear. Imagine your sample has these 8 different viruses floating around in it. How is a PCR test supposed to pick out the one that's avian influenza? And the answer, according to the source, you actually can't be 100% sure. A test that's just looking for one short snippet of genetic code could get triggered by any of these viruses, as long as they happen to share that tiny little sequence. So theoretically, they could all light up as positive. Okay, so this is where it all comes together. This brings us to the source's main case study, where it applies all these arguments, the whole situation surrounding highly pathogenic avian influenza, or HPI.
The author takes this critique and points it directly at HPI. They claim that these PCR diagnoses are what's leading to call orders for animals. But they also point out that only about 10% of these infected cattle even show minor symptoms. As for human risk, the source really downplays it, noting the one U.S. case in a farm worker was just pink eye. So the bottom line is, it questions any research on HPI that's only using PCR results, without actually finding and isolating a complete live virus first.
And just to really drive home that point about human risk, the source gives us this visual. You've got hundreds of thousands of farm workers in the US on one side, and on the other, four reported infections. The The source presents this as a huge gap between the raw data and how the risk is being perceived.
Now, the author of our source didn't just write all this down and hit publish. They actually tried to get some answers. As they lay out in their article, they reached out directly to the official regulatory agencies over in the United Kingdom. And this timeline here documents that whole back and forth. The author emails the UK's VMD and asks, hey, who validates these PCR tests? The VMD writes back and says, not us, you need to talk to the APHA. So the author does. They forward the email to the Animal and Plant Health Agency. After a bit of a wait and a follow-up, the APHA finally replies that, PCR technology has moved on since it was invented. When the author presses for more specifics, the final response they get is basically, we're done talking about this. The correspondence is closed. So the core questions were just left hanging in the air.
So where does that leave us? After breaking down the science as the source explains it, looking at all the core arguments, and seeing this documented search for answers, it all comes down to one final, really big question. And this is it. This is the question at the very heart of the source material. When a test result can lead to these massive consequences, we're talking about culling entire herds of animals or shaping major public health policy, who is on the hook for how that test is being used? Who is ultimately responsible for making sure it's appropriate? It's a really provocative question and a powerful one to end on. Thanks for joining us for The Explainer.